The overall goal of this study is to use digital holographic microscopy, or DHM, to provide automated stain-free quantitative phase contrast imaging of intestinal inflammation in colonic tissue samples for the multimodal assessment of epithelial wound healing in vitro.
This method can help answer key questions in the field of gastrointestinal inflammation and wound healing about the disease activity in inflamed human and marine colon in situ.
The main advantage of this technique is that it allows the detection and quantification of cell and tissue biophysical parameters in a stain-free and objective manner. Demonstrating the procedures will be Steffi Ketelhut, a technician from the Biomedical Technology Center.
Set up the cell culture. Incubate 4 × 10⁵ Keiko-2 cells per centimeter squared in 35 mm petri dishes equipped with high culture inserts in RPMI medium, supplemented with FBS and antibiotics at 37 degrees Celsius in 95% humidity and 5% carbon dioxide. After 2 days, treat the cells with the reagent of interest and return the cultures to the incubator for an additional 24 hours. The next day, aspirate the supernatant and use tweezers to remove the inserts. Wash the culture 1 to 2 times with 1 milliliter of Heapy's buffered cell culture medium to remove any dead cells and remaining cellular components from the wound area. Then, add 2 milliliters of fresh Heapy's buffered medium and close the dish. Now, place a petri dish.